Good morning. Thank you all for being here. Welcome to Berkeley, welcome to the University of California, to the Innovative Genomics Initiative, and to the second annual CRISPR Workshop. So we did this workshop last year, as Jacob implied, but this year we've added a lab component. And you are the guinea pigs for the lab component. I hope everything goes smoothly. If it does go smoothly, I want to acknowledge Emily Lingeman and Amos Liang in particular, who are a student and a postdoc here in Jacob's lab, who have worked extremely hard to make sure that everything that we need for the lab component is there, functional. And if it fails, blame me, okay? I will be here all week, so if there are things you want to say about the structure of the workshop, about the content of the workshop, about any of the logistics, you're welcome to come to me. Marcia and Diane may know more about the logistics than I do, and you will have TAs in the lab who will help you through the laboratory experiments as well. So I just wanted to start by going over the schedule briefly. And let's see, let's hope this is on. Okay. We're here. We're going to have lectures in the mornings, a couple of lectures by me on sort of basics of the CRISPR system. Luke Gilbert will come in and give a talk about, beginning to talk about applications of the dead version of the Cas9 protein. We'll have lunch, and then we'll go over to the lab. The lab is in a different building. You have a map in your packet. If you look at the map, we're down here sort of in the bottom left corner. The lab is in the building that's just northeast of us a little bit. It won't take long to get there. The other thing that's highlighted is the faculty club, which is where we'll have dinner on Thursday evening, okay? At 1:30 this afternoon, after we've had a chance to have some lunch, I'll lead a group from here over to the Valley Life Sciences building where the labs will take place. You can get there on your own if you want. There's no penalty for doing that. But if you'd like to go over with the group and make sure you get to the right place, we'll be leaving here right around 1:15 or 1:20, okay? What does the building look like? It's a
giant white marble building.
Not the only one on the UC campus, however. But it's kind of a neat old building. It has engravings that say zoology and biochemistry and plant biology, things up top. Anyway, it's pretty easy to find and it's close. So then there'll be lab experiments. When you get to the lab, we will first of all break you into groups. The lab is set up so that there are 6 different lab benches, each of which can accommodate 8 people. We may not have quite that many here, but we'll assign you to particular lab benches. This is important because at each lab bench, each person will be given oligonucleotides that will lead to the synthesis of 2 different guide RNAs for 2 different genomic targets. It's important that you stay at that lab bench with that lab group because the ultimate analysis depends on our knowing which targets you're attacking. So stick with the lab group you're with, and we'll do that assignment once we get over there. You'll also get an introductory description of some more detail about what's going to be going on in the lab from one of the TAs each day. Okay. On Tuesday, we'll have more lectures, one from Luke Gilbert, more extensive discussions of applications of the dead Cas9. Martin Kampman will talk about screening with the CRISPR system. The lab on Tuesday is going to be the one where we actually make ribonucleoprotein and get it into cells. And that day, the lab is going to be a little bit chaotic, I'm afraid. but I think we've got things more or less under control. It may take a little longer than the other days. It's the only day where I think we may have some bottlenecks in some of the cell handling, but we'll try to work everybody through that. Okay. On Wednesday, we have some more sort of basic talks about informatics relating to applications of the CRISPR system. And then we have the first laboratory application, which will be discussed by Dirk Hockemeyer, who's a faculty member here, and he'll talk about applications to stem cells. We have some time left for open discussion if you want to talk about things that have happened. If there's not much interest in discussing things that have happened up to then, you can have a little free time. And each day we'll have breakfast starting at 8, and then lunch either at 12 or 12:30, depending on the day. That afternoon, this is a dry lab exercise that Martin will lead along with Max Horlbeck and a couple of other TAs. So if you brought your laptop computer, please bring it to the lab. If you didn't bring a laptop computer, you can share with somebody else at that time. On Thursday, we have more talks about applications to different systems. I think this is going to be really exciting. These are people who are sort of pushing the envelope in each of these research areas. And some of them are leading to more societal applications as well as just doing research. The lab that day is simply harvesting cells and preparing genomic DNA, amplifying the targets. And that evening, we'll have dinner at the faculty club. There's a reception and then dinner. The lab may not go all the way till 5:30, but we'll gather by 6 o'clock over at the faculty club. I'll just go back for a minute and say that this afternoon, after the lab, we'll have a reception here, the lobby on the second floor of this building at the east entrance to the building. And several of you have brought posters, and I'm grateful for that. So this will give you an opportunity to see what some of your colleagues in the workshop are actually working on and what their potential or current applications are for the CRISPR technology. So that's 6 to 7. There will be refreshments, adult refreshments, and a little bit of food. And then where were we? We're up to Friday. Friday, Mike Holmes from Sangamo Biosciences, which is just up the road in Richmond, is going to come and talk about some of the clinical applications that they have been pursuing of the genome editing technologies. And as you probably know, Sangamo has focused on zinc finger nucleases, but it's a good model for what's going to be done eventually with the CRISPR system as well, and give you some idea of how these tools are making their way to the clinic. Then Hank Greeley, who's a professor of law at Stanford University, is going to come and talk about ethical, legal, and societal issues. And Jonathan Weissman from UCSF is going to continue that discussion. Jonathan's a member of the National Academy Committee that's doing a very broad survey of what are and how should we deal with these ethical, legal, and societal issues. So I think that'll be quite interesting to you. Now, as a wrap-up, you get to see whether your guide RNAs worked. You get to see whether you're actually now experienced CRISPR genome editors, because we will do the final assay on the genomic DNA from the cells that you've targeted. And then the workshop's over. So I hope you have fun during the week. I think you'll find it's a nice mixture of lectures that'll teach you about things that other people are doing, and then you have the labs to actually do it yourself. And as I say, I'll be here all week if you want to chat with me about any aspect of the workshop. Any questions about logistics? Yes?
Can we post photos of the lectures or labs on Twitter?
I don't see why not. We're videotaping all the lectures, and they will be posted after the workshop. Yeah, I know Marcia has sent you messages saying, feel free to tweet during the workshop. Try to keep it polite, you know. But yeah, if you want to, if you have a Twitterverse you want to let know about what you're doing this week, feel free to do it.
There's an IGI Twitter account at IGI-SOC you can tweet to, and there's a hashtag that Marcia set up, just hashtag CRISPR Workshop.
Did you hear what Jacob said? Okay. Other logistic issues? All right. So I'm going to launch into this lecture. So what I'm going to do is I'm going to talk a little bit about some of the history of genome editing tools, and then a lot about aspects of the CRISPR system itself. After the break, I'll talk about, I spent a lot of time talking about off-target effects, delivery of the Cas9 and guide RNA reagents. But here we go. Ready? All right. First thing I'd like to say about genome editing is that nature's been doing it for a really long time. So this is just a picture of Darwin's finches. He realized that these different closely related birds had beak sizes and shapes that were adapted to their mode of feeding. And these arose by natural selection amongst naturally occurring genomic variants. So natural selection is a form of genome editing. Humans got involved in genome editing when they began agriculture, selecting crops and livestock. So this is teosinte. It's the precursor to our current corn. And it was just by humans picking plants that had favorable characteristics, grew bigger, had bigger cobs, larger seeds, that got us to the point where we have the corn that we feed to animals, people, and gas tanks these days. So geneticists in the early days relied on naturally occurring sequence variants in order to study inheritance of particular characteristics. And then in the first half of the 20th century, it was discovered that by using ionizing radiation or chemicals on Drosophila that you could enhance the frequency of mutations. And so now geneticists could perhaps work a little bit faster because they could make new mutations, but they were still essentially random. And it was only getting into the late 1970s into the 1980s that it became possible to make targeted mutations so that you could identify a gene you were interested in and actually modify that particular gene. And this was done initially in yeast, but then this is my colleague Mario Capecchi figured out a way to do this in mouse cells, and in particular in mouse embryonic stem cells, so that you could generate genetically modified mice out of cells in culture that you had manipulated at a specific site in the genome. And this required, in both yeast and in mice, a very powerful selection scheme. This is the one for mouse ES cells, so that you could identify the cells that had enjoyed the genomic sequence change that you had tried to introduce. The reason this didn't go beyond yeast and mice is that the efficiency of getting the DNA that you manipulated in the laboratory into the genome was terrible. It was about 1 in every million cells, and sometimes considerably worse than that. So it took this very powerful selection. But information accumulated over a period of time had made it clear that the problem here was that the target for this gene targeting experiment was essentially inert. There's no reason for a continuous stretch of chromosomal DNA to engage in homologous recombination, which is what's required to get that piece of DNA to replace the endogenous sequence. But if you can make a break in the target at the same time that you're providing the donor DNA, then the frequency of recombination goes way up. And that makes a tremendous amount of sense because a double-strand break in chromosomal DNA is potentially lethal damage, and homologous repair is one of the ways that cells go about fixing that damage. So how did we learn that double-strand breaks stimulate recombination? Well, it's a combination of things coming together from research in a variety of areas. One aspect was radiation damage can stimulate sister chromatid exchanges in chromosomes in cells. And this is a little hard to see, I think, but these are chromosomes from cells that have been differentially labeled in the sister chromatids. And you can see that the yellow and orange segments have occasionally exchanged along the length of these chromosomes. The frequency of that is greatly stimulated by ionizing radiation, one of the products of which is double-strand breaks. Another place that this information came from was studies of meiotic recombination. We now know that meiotic recombination in essentially all organisms is initiated by intentional double-strand breaks that are made during meiosis by a protein that in most organisms is called SPO11. So intentional double-strand breaks are required for the crossing over that leads to proper chromosome segregation during meiosis. And then in the late 1980s and into the 1990s, people did experiments with these nucleases. They're called meganucleases typically. They're natural DNA endonucleases that have long recognition sequences. And so Jim Haber using HO and Bernard Dujon-Maria Jason using the meganuclease called ISCE1 showed that if you put the recognition site for one of these nucleases into a mammalian chromosome, that introducing the enzyme that breaks that sequence stimulates localized recombination. Okay? So all that information came together to say, well, what we need to do in order to make gene targeting more efficient is to make double-strand breaks in the target. So we needed a method to make double-strand breaks, but it also has to, the reagents for this method had to be programmable so that we weren't limited to a single target as you were with HO or ISCE1. It had to be programmable so that you could attack essentially any target that you want. And as you probably know, there are sort of three platforms that are in use that fulfill the pair of criteria. The zinc finger nucleases, the TALENs, and the CRISPR-Cas system. And I'm going to tell you a little bit about how each of these came about. At my age, you get to tell history. You get to tell the war stories. And I'm here and you're sitting in the audience, so if you walk out, I'll know you're not interested. But I think these are sort of interesting stories. So the zinc finger nucleases were the first of these programmable nucleases that emerged. And it came from studies on the FOC1 restriction enzyme, which Chandrasekharan and his colleagues had demonstrated had separable DNA recognition and DNA cleavage domains. They could separate them either by partial proteolysis or by cloning. And so they said, well, what if we take the nuclease domain of FOC1, which doesn't have any sequence specificity itself, and we link it to a different DNA binding domain, which will provide the sequence specificity. Can we direct FOC1 cleavage to an arbitrary site? And they tried several different DNA recognition domains, but the one that got us excited was when they linked the FOC1 cleavage domain to a set of zinc fingers. Zinc fingers had been identified as modules for DNA recognition that exists naturally in a wide variety of eukaryotic transcription factors. So putting these two things together, studies on FOC1, studies on eukaryotic transcription factors, led to the generation of the first zinc finger nucleases. And my lab collaborated with Chandra's lab to define what was required for cleavage by zinc finger nucleases. Like the natural enzyme, the cleavage domain has to dimerize in order to actually cut DNA. A monomer is not a nuclease. Because the dimer interface is very weak, in order to get efficient cleavage, you had to have two sets of zinc fingers, each linked to a monomer of the cleavage domain in order to get cleavage. And this was actually very beneficial because neither monomer is by itself a nuclease. It's only when the two monomers come together by binding sites that are very close to each other that you actually get target cleavage. And so this minimizes off-target cleavage. We've studied the relationship between the spacer between the binding sites and the linker in the protein. We showed that both cleavage and recombination could occur at high efficiency in a eukaryotic cell, even when the substrate was wrapped up in chromatin. And I think this was quite important because the nuclease domain comes from a bacterial enzyme. And it would never in its natural life have seen a chromatin target. But this worked quite well. And of course, we began changing the specificity just by changing the identity of these zinc fingers. And an important feature of this system is that recognition can be manipulated separately from the cleavage activity of these proteins. And in fact, people have manipulated this in various ways without changing recognition for beneficial properties. So, starting in 2010, 2011, people began using what we now call talons. And they're a lot like the zinc finger nucleases, except that, since they use the same FOC1 cleavage domain, but the DNA binding domain is made up of modules, each of which recognizes a single base pair. The zinc finger modules each recognize 3 base pairs, but these TAL modules each recognize a single base pair. So where did they come from? Well, they came from proteins made by bacteria that infect plants. And the majority of the species that have been studied that have these transcription activator-like effectors, or tails, the majority of these have been identified in the genus Xanthomonas. But a lot of plant pathogens produce proteins like this. And these proteins are produced in the bacterium, they're secreted into the host cell, and then they go to the host cell nucleus and regulate host genes in ways that promote the infection. And when enough examples had been studied of these odd repeats in the tail proteins and the targets that they bind to, it was learned that each of these repeats has a framework of about 32 amino acids that don't change much from one repeat to the next. But an additional 2 amino acids in stereotype positions called 12 and 13 vary in concert with the base pair that's being recognized in the target sequence. And by looking at a bunch of these, you could actually develop a code of recognition. And the one that's boxed down here from Adam Bogdanov's studies is the one that's typically used even now. When those 2 amino acids are a histidine and an aspartic acid, that module recognizes a C in the DNA sequence. When that's an asparagine and a glycine, T is recognized, etc. The module for G actually recognizes A in some contexts almost as well, but it's still pretty good. And there are alternatives for G, like this one, which instead of having 2 asparagines, has an asparagine and a lysine that's more specific, but provides a little less affinity. But there's a code, and so it's really easy to assemble these talon proteins to recognize new targets. We're now, of course, in the midst of the CRISPR craze. nobody here I can really tease with this, that sometimes I say the CRISPR craze is a dance, and the TAs will be doing this in the lab, that they aren't here right now. So where did the CRISPR system come from? Briefly, in the 1980s, a group in Japan was sequencing a gene in the E. coli genome, a particular strain of E. coli, and stumbled on to these strange repeated sequences. In this array, there's a repeated sequence, this black box, which occurs over and over, and between the repeats, there are what are called spacers that differ at each position between the repeats. 15 or 20 years later, people recognized that these spacers correspond to viruses or transposable elements or plasmids that these particular cells would be exposed to during their normal life. And it was proposed that perhaps this constituted an immune system, an adaptive immune system, where these sequences from a virus infection, for example, provided a memory that the cells could use to combat the next time a virus of this same class tried to infect the cell. And systems like this have subsequently been identified in, I think what people are now saying is 90% of all archaeal organisms and about 50% of all species of bacteria. So it's very widely distributed. So how does this work as an immune system? The cell transcribes this cluster of repeats and spacers into a long precursor RNA. That precursor is chopped up into smaller pieces, each of which contains part of the spacer and part of the repeat, through the agency of a protein called Cas9, another RNA called tracer RNA that hybridizes to each of the repeats, and with some participation by RNase 3, cellular RNase 3. The product of that processing is a collection of Cas9, CRISPR RNA, tracer RNA complexes. And each complex represents a different one of these spacers in the original cluster. Then when a virus infects, if the cell has a spacer sequence that corresponds to a sequence anywhere in that viral genome, it directs Cas9 to that sequence and leads to cleavage of the viral genome and inactivation of the infection. Okay? And what people have done now, what we do as experimenters, is just to take advantage of this part of that whole system to make targeted double-strand breaks in essentially any DNA we want to. And I thought I had another picture of Cas9 here, but I guess not. It was studies that were published in mid-2012 that identified the necessary components of this system. And you can just see here, this is using plasmid DNA as the target. Here's the plasmid DNA, Cas9 protein, a CRISPR RNA that corresponds to a sequence in that plasmid DNA, the tracer RNA, and the cleavage depends on magnesium. If you have all those components present, then you get cleavage of this plasmid DNA. So this is the uncut plasmid, supercoiled and relaxed circles, and after treatment, you get the linear DNA. And linear DNA can also be cut into smaller linear molecules. Oligonucleotides can also be cut with that same combination. And if you leave out any of those components, the cleavage doesn't happen. Depends on Cas9, CRISPR RNA with a sequence that's in the target, and the tracer RNA. And up till that time, nobody knew that the tracer RNA was required for cleavage. And this is a picture of, a diagram showing tracer RNA that has complementarity to the repeat part of the CRISPR RNA, and this is the part of the CRISPR RNA that corresponds to the target. And the cleavage sites could be mapped on both strands of the target. And these materials were derived from Streptococcus pyogenes, and we're going to talk about CRISPR components from other systems as we go along. The same group, this is Martin Jinek, Christophe Chelinsky, Emmanuel Charpentier, and Jennifer Doudna. I think there were one or two other authors on the paper. They also looked at the sequence. Other people had done a portion of this as well, and saw that there were sequences that corresponded to two known nucleases. They're similar to two known nucleases, one of the HNH family and the other of the ROV-C family. And what they could show is that if you inactivated active site residues, if you mutated active site residues in either the ROV-C domain or the HNH domain, you could only nick a plasmid instead of making a double-strand break, and that each of these modified enzymes cut one strand. This cut the complementary strand, or the other. This one cut the non-complementary strand. So the two nucleases in Cas9 each cuts one of the strands of the target DNA. And finally, in this paper, they showed that you could simplify the system by making a chimeric RNA that has the essential features of the CRISPR RNA and the tracer RNA. So the tracer RNA seems to be required for binding the CRISPR RNA to the Cas9 protein, but not all of the tracer RNA is necessary. And they showed that both in plasmid cleavage and in oligonucleotide cleavage, that this chimera A was adequate to make double-strand breaks. The chimera B with a shorter extension of tracer RNA didn't work as well, and frankly, now people use an even longer version, has a little bit more of the tracer sequence here. It's a bit more efficient. Okay? So that set up the system, and here's a simplified version of it. In order to cut something in the target, you need a guide RNA. We often call this a single guide RNA that has a sequence that matches the target. Typically, this is 20 nucleotides of DNA-RNA hybrid. There have to be sequences that came from the remainder of the CRISPR and tracer RNA that help binding to Cas9. You have to have the Cas9 protein. And Cas9 also requires a short sequence just downstream in the way we typically draw this from the DNA-RNA hybrid in order to recognize the target sequence. For the Streptococcus pyogenes Cas9, this is any base followed by two Gs in that top strand. Then those two Gs are not in the guide RNA, they're only in the target. And a key reason for requiring a PAM sequence is that the CRISPR RNA, when it's in the genome, provides a target for cleavage, except it doesn't have the PAM sequence. And without the PAM sequence, without having the PAM sequence in these genomic copies, you won't get cleavage of the bacterial genome. So it's a way to protect the genome. So why do people now use the CRISPR system all over the world, love the CRISPR system? What advantages does it have over its predecessors? Well, it's really simple to design new RNAs for new targets. You just have to know the Watson-Crick base pairing rules. You could assign this to a junior high school student and have your answer in a couple of minutes. This is maybe the most important feature. There's only one constant protein that's required. For the ZFNs and talons, you had to redesign two proteins for each new target, which was time-consuming and didn't always work exactly the way you wanted it to. But here there's just one protein involved. It's very easy to produce the guide RNA and the Cas9 protein. You'll produce the guide RNA starting today. doesn't cost very much. You just have to have basic molecular biology skills. Because there's only a single protein involved, by providing multiple RNAs in some format, you can multiplex the system. And there are many variants of this that are now available, and I'm going to tell you about quite a number of them. Okay, before going on to some more aspects of applications, I just want to say that these tools came from completely unexpected places. Nobody set out in studying eukaryotic transcription factors to find a DNA binding domain that would be used in genome editing. Similarly with the Xanthomonas tail story and the CRISPR story, it started out just as odd sequences in some bacterial genomes. But as time went on, various threads came together to turn these into very effective genome editing tools. And that's how science goes, right? If you don't study how the world works pretty broadly, you'll never come up with anything that's really new and useful. Okay, so we have the ZFNs, then chronologically the TALENs, and then the CRISPRs. So what do these magical reagents do? All they do is make a double-strand break in DNA. That's it. So even the CRISPR system is only a nuclease. Well, fortunately, it's a very targetable nuclease, but it doesn't do anything else. Everything that happens after the double-strand break is made depends on cellular DNA repair activities. So the cell recognizes double-strand breaks as potentially lethal damage and goes out of its way to fix that damage. So if we make a single break in a single chromosome in a complex genome, the cell would normally either use the sister chromatid for a homology-dependent style of repair, like the gene targeting that we were talking about before, or it'll jam the ends back together using non-homologous end joining. We can introduce a template for repair, as I suggested with the gene targeting discussion, that the cell will use as an alternative to the sister chromatid for repair. And this will allow us to make sequence replacements of our choice at the broken target. Even when there is a donor around, and certainly when there's not, the cell will jam the ends back together. Often, the ends are joined accurately. There's no mistake made. But if that happens, that target can be recut. And occasionally, the cell will put those ends back together inaccurately, making localized small insertions and deletions at the target. So these are the cellular processes we depend on as researchers to make the modifications in the genomes that we're working with. So let's talk first about non-homologous end joining. What kinds of mutations are produced? Well, insertions and deletions, we typically call these indels. That's a neologism. The CRISPR system favors deletions over insertions, but you still get insertions, sometimes with a deletion. Some of the insertions are flanked by microhomologies. What do I mean by a microhomology? Well, if you make a break at a particular site in a genomic sequence, sometimes that genomic sequence has short sequences to the left and to the right of it that are the same. And when an exonuclease gets on at the ends at the break and chews back a little bit, it can expose single strands that contain from one side one strand and from the other side the other strand that match. And so here's ACAGC, ACAGC, and over here at this particular target, ACAGC. And so if you get the chewing back of one strand and this ACAGC gets together with the GCTGT over here, you can form a short duplex like this that directs repair. it's a non-homologous or a micro-homologous repair process that sets the register for how the repair is going to happen. And I have to say that we don't know in detail how that repair ultimately occurs. I have a favorite model that we can talk about at some point if you like. But Jinsoo Kim's lab set up a scheme for evaluating whether any particular target would have favored microhomologies. They can be as short as 2. Sometimes we even account a single base match as being a microhomology that might have directed the repair. Whether that's actually how that occurred or not, a little hard to tell. But what they did, in addition to just saying whether these microhomologies existed around any particular break, was to look to see whether, if they were used, they created an in-frame or an out-of-frame deletion, if this was in a protein coding sequence. And if you were to make a break where there was a good microhomology that led to an in-frame deletion, you might choose a somewhat different target so that you were assured of getting out-of-frame deletions using these favored microhomologies. And that's just what's shown here. A break here has a pretty long microhomology that would lead to out-of-frame deletions. But this one, if you cut over here, has a long microhomology that would lead to in-frame deletions. So you might want to choose the CRISPR nuclease that cut here. The guide RNA led to a cut there, rather than the one that led to a cut over here. So microhomologies. Just before we leave the non-homologous end-join products, I want to tell you about one we stumbled onto in Drosophila many years ago. So this is a really strange insertion. The insertion was 720 base pairs long, an abnormally long insertion. 704 of those base pairs corresponded to another gene in Drosophila. So the target gene into which this was inserted was the rosy gene, the gene we targeted over and over. But 704 base pairs of this Gilgamesh gene were inserted. But it wasn't sequences from the gene per se. It was actually sequences from the first exon and the second exon, and the intron was cleanly gone. So at some point, whether directly or indirectly, a processed messenger RNA, a spliced messenger RNA, provided the template for insertion of this long sequence into the target that we were cutting. So you get some pretty weird insertions sometimes. The other thing that's characteristic of these insertions of various kinds is that you often get sequences inserted that you can't really account for. So despite the fact that most of this insertion came from the Gilgamesh gene, there were 7 base pairs on one side and 9 base pairs on the other in the product that didn't correspond either to the donor, Gilgamesh, or to the target, Rosie. And so I call them space dust. Where else would they have come from, right? Anyway, just to say that we don't have control over the end-join mutations, although looking at microhomologies and so forth, we can bias things a little. Yeah?
I can't resist. I want to share a similar experience that I had. Actually, a co-worker of mine found this sequence, but it looks as though the double-strand break was interpreted by the cell as a telomere, because we saw this repeat sequence. It was about 200 bases long. It was a 6 nucleotide repeat, and we're looking at it, and she's saying, the sequencer is broken. It just went through this 200 nucleotides and then trailed off. When we Googled it, because Google is such a great genome editing tool, right? And found that it's the signature 6 bases.
Of the,
It's on the end.
as of the telomere.
So yeah, space dust. In this case, we were luckily able to figure out.
Yeah. So
Strange things happen.
another strange insertion. We also found insertions that corresponded to a histone gene and a ribosomal RNA gene. We couldn't tell in those cases whether RNA might have been the template because they didn't cross an intron. But it's plausible since those are both abundant RNAs. And anyway, thank you. So there's some strange stuff that goes on that we don't have complete control over. I'm going to go through a few methods of detecting end-join mutations. These methods will also detect insertions that came from a donor DNA, depending how that donor was designed. So this one is HRMA, high resolution melt analysis. Most of these methods depend on PCR amplification of the target and then some sort of downstream analysis. This particular one depends on the fact that you will have made mutations that differ in sequence around the target from the pre-existing wild-type sequence. And so if you denature and renature these PCR products, some of which are mutant, some are wild-type, you'll get heteroduplexes and homoduplexes, and as long as these DNAs are short enough, they will melt at different temperatures. And this is high-resolution melting. This whole scale here is just 10 degrees. And this is a real example. This is from a paper by Tim Dahlem and colleagues, some of them at Utah, showing taking zebrafish embryos that have been targeted with a particular pair of talons and just doing this HRMA analysis on the target that they were going after in mixed samples. And so you can, the wild type will melt with this unique melting curve, but the heteroduplexes and some of the mutant sequences will melt at different temperatures. And so you get, because this is a mixture, you'll get a lot of different melting curves from different embryos. This was a pair of talons that worked quite well. There's very little wild-type sequence. This was one that didn't work so well. There's not so much wild-type sequence. So this doesn't tell you what the sequences are, but it tells you whether the targeting worked and you got some sequence alterations. More commonly, people will use mismatch-dependent endonuclease assay, and this is what you're going to do in the lab on Thursday and Friday. So if you introduce nuclease into cells, let the cells go for a while, something happens behind the fig leaf, and you just want to know if something good happened. So you isolate genomic DNA, amplify the target, you'll get you'll get homoduplexes by amplification, but there'll be different sequences there. So if, as in the HRMA example, you denature and reanneal, you'll have some homoduplexes of various kinds and heteroduplexes. The T7 endonuclease, called T7E1, will only cut at mismatch sites. And so if you take this mixture, treat with T7E1, and then run a gel, as you will do, you will see something like the following. These are very good guide RNAs. So you take that PCR product and you don't treat it with T7E1, and it runs as a nice tight band. You treat with the enzyme, and it cuts a lot of those into smaller fragments. And there's another band down here. It's a little hard to see. These are two examples of a very similar guide RNA, and this is a different guide RNA. And in the control, even treating with T7E1 gives you no bands down here. So this is an indication of successful cleavage, at least, and probably non-homologous end joining, although depending how the experiment was set up, there might be some homologous products in there too. I like to show this. This is kind of hard to see with the front lights on, but that's an agarose gel. This is some of the same samples on a polyacrylamide gel. So here, let's look at this sample right here. Here, prior to cutting with T7E1, we've got kind of a smear and a bunch of stuff up there. So what's that? Well, those are heteroduplexes. They run bizarrely on a tight polyacrylamide gel, I think because they're sort of floppy and they get hung up on a tight matrix. In an agarose gel where the matrix is much looser, this doesn't happen. So I think it's sort of fun to run these things on a polyacrylamide gel and kind of see this without even doing the cleavage with the endonuclease. But after cleavage, you get things that run down into the gel like we saw before.
Are these methods also useful to analyze a clonal derivative of a, because in that case, let's say you get a perfect, you get basically both alleles to the same type of lesion, and this won't show up.
That's right.
At all, right? So in that case, are there other better methods to do, or if you get a mixture of two alleles, like one is cut, the other one is not?
Yeah. So the things I'm describing are really bulk analysis. You take a population of cells or an organism that you've treated, and you do the analysis in these bulk ways. If you want to know cell by cell or organism by organism, probably the best thing to do is just sequence. You can amplify the target and sequence it if you've got a clone of cells. If you have a clone of cells, you can do whole genome sequencing and see whether that's the only thing that happened during the treatment of the cells. You can look to see if there are other changes in the genome. We'll talk about that a little bit later. Don't remember when I'm supposed to stop.
Type sequence too.
Pardon?
You can add in some wild type sequence. And if you generate a clonal mutant, the presence of the wild type will generate produplexes when they're mounted.
Right.
You can screen.
Right.
That's one thing we've done.
Yeah. Okay. Another method that some people are quite enthusiastic about is called TIDE, and I don't remember what it stands for, but you start the same way. You just PCR amplify the target. Then you feed it to your local Sanger sequencing facility. And the wild type sequence gives you these nice clean reads. And as you're sequencing the PCR product, you're getting a matched, very clean read until you get close to the point where the cut was made. And then all of a sudden, things start going to hell. So if I remember correctly, up till this point in this sequence, you got CCG, and then here where you're supposed to have a C, you have some Cs and some Gs, and everything downstream from here is a mess. Now, if you were sequencing a unique fragment of DNA, you think, oh, I screwed up, I got to throw this away, I got to try again. But here, there's you can use an available computer program to do decomposition. And I don't know how this works, but you get an output that shows you the frequency of deletions and insertions of different sizes that must have been present in order to give that gamish. Okay? And so this has the advantage that once you've done the PCR, you don't do another experiment yourself, right? You just send it to the sequencing facility, and then run this analysis on the sequencing products. They claim that it is semi-quantitative. It doesn't give you the sequences. So if you're really interested in the sequences, I mean, what they show you here is the percent of all sequence that is represented by that's probably a -4 deletion. And it says, 15% is a -4 deletion. Whether that's the same -4 deletion or not, it's not clear, okay? All right. Another thing you can do is whole genome sequencing. You take that PCR product that came from a population of cells and you send it off for next generation sequencing, and then you hand the results off to a bioinformatics expert, and he can tell you, based on comparison to the wild type sequence, how much wild type, how much indel, what proportion of indel sequences are present. Just if we look over here, this is getting close to 50% indels. And since you know the sequence of the donor DNA that you put in, this analysis can also tell you what proportion are representative of incorporation of that donor sequence. And so this is results of experiments that Mark DeWitt and I were involved in when I was here on sabbatical last year. So if you've got next generation sequencing available to you, it gives you lots and lots of data that you can coalesce into answers like this. One of the frustrations for me about the next generation sequencing was I couldn't see everything. I could just see the summaries. And it's quite difficult if you have millions of sequences and there are sequencing errors involved to sort everything out so that you can see all the individual sequences and how often they're represented. It's possible, but it's not trivial. All right, let's talk about homologous repair for a little while and generating donors. And let me just say in summary that if you're using long double-strand donors, for whatever reason, you may be using a long double-strand donor because you're trying to insert a pretty big sequence. It may be because you've already got a plasmid that carries the sequence you want transferred into the genome. You need typically, and I'll show you a counterexample or two. Typically, if you're relying on homologous repair, you would want to have a few hundred base pairs on either side of an insertion, on either side of the break. A total of something on the order of a kb of homology. Whether you use a linear or a circular donor depends to some extent on the biological system you're working in. We found in Drosophila that if you generate the donor in situ by cutting it out of someplace else in the genome, the linear worked great. But when we started doing things by embryo injection, the linear donors were totally ineffective. I can hypothesize why that is, but circular donors worked much better. So depending on the biology of the system you're working with, that will determine what the best configuration of the donor is. Single-strand oligos work well in most systems. As long as you don't have too much to change, you can just use an oligonucleotide to make that change. Even as short as 30 nucleotides total with homology feet on either side of the break is enough to get a fairly good level of homologous repair. You have to have homology right at one end of the break, and I'll show you an example of that in a moment. And typically we design our donors so that they introduce sequence changes that prevent recutting. So if you get a homologous repair event that puts in the sequence you want, you don't want that cut again with the danger of end joining making indels after you've put in a nice clean sequence. So that's something that we typically do, although it's not absolutely required. All right, what about this homology to one end? This represents experiments we did quite some time ago in Drosophila, where we were introducing large insertions or deletions. So here's the cut site. The standard donor has mutations that prevent recleavage, and you get good incorporation. If we incorporate deletions that are off to one side or the other, or go from the cut site just to one side, all of those work, as does a big insertion. This is, I can't remember, a 5 kb insertion or something like that. Those all work pretty well. But when we tried a deletion that covered the cut site and deleted out on both sides, we never got any products. And I think the reason for that is that the way homologous repair is initiated is from one of the ends at the break. So the target is actually invading the donor. And if there's no homology to an end at the target in the donor you're trying to incorporate, you can't get a homologous repair event. So this is one important thing in donor design. to make sure that you have homology that's pretty much right up against one of the ends at the break. Now, something like this works because this end finds homology, and then it's copying from there, and it just copies across that deletion.
You mean it immediately at the deletion, not internal?
Right. So
you have homology internal of the actual ends of the DNA. That doesn't work as well. Is that what you're saying?
I don't know how far in you can go. 2 or 3 nucleotides, that's fine. But there just isn't much removal of nucleotides from the 3' end after a break. And so if you got both ends chewed way back, so you cut here and you chew way back, you'd run into homology on this donor. But that doesn't seem to happen if you're more than a few nucleotides away. At least that really decreases the efficiency. Okay? All right. So what about incorporating sequences that are some distance from the break? We studied this in some detail in Drosophila, and this is just showing that we had a donor and a target sequence that had a number of polymorphisms that we could monitor. And we found that these conversion tracks were very broad. This reflects how far from the break do we get incorporation of sequences from the donor. And on both sides of the break, this has been extended out even farther. On both sides of the break, you go 3 kb away, and in Drosophila embryos, you're getting about 50% incorporation from the donor compared to the amount you're getting at the target site, which has to be repaired in order for this to be homologous repair, right? Sort of complicated sentence. And it was a little bit different in when we induced the nuclease in larvae. The discouraging thing is that in mammalian cells, these conversion tracks are really short. So you have to make a break close to the site where you want the donor to introduce sequence in order to have it done efficiently. Right? So there's another lesson.
Have you tried simultaneously cutting 2 distant places?
Yeah, that works. I'll show you some examples of that from other people's work. So here at the IGI, Chris Richardson, who's a postdoc, thought maybe he could improve the incorporation of oligonucleotide donors by thinking about mechanism and looking in detail at how Cas9 makes a break. So unlike the ZFNs and talons, Cas9 makes, it handles the DNA strands in an asymmetric fashion. One strand is bound by the guide RNA, and so it's important that Cas9 recognize that there's a good duplex here as well as some of the other features. But the other strand, at least to a first approximation, is just displaced. So maybe it's more available for initiating recombination than the other strand. And so he played around with oligo-donor design. Both strands work. This is where the cut site is. Both strands work. This is the percent incorporation of donor sequences at the target. But with some donor designs where they're a little bit asymmetric, you get quite a distinction between a donor that's complementary to the displaced strand and a donor that's complementary to the RNA-bound strand. And you can see that in 2 examples here, where the green donor is better than the red donor, green donor is better than the red donor. And this doesn't give you an order of magnitude. It doesn't work for every particular donor design, but it's something that is available to try, and I've given you the reference to the paper. By the way, the slide presentations will be posted on the workshop website, mine at least, after the talks. Yeah.
This is the percentage for all cell type or just for this?
I don't believe this has been tested in a lot of different cell types. I don't know that a lot of people have actually tried it. You know, there are tweaks to the system that are out there that people haven't used even though they sound like they're good in principle, because the basic system is good enough for a lot of things. Now, and this paper was just published early this year, so I don't know how many people are aware of it. So just another thing to think about. So here's some of these two-cut approaches. So there have been papers from Geraldine Saidou's lab for a couple of years now talking about using donor DNAs with very short overlaps. So the donor DNAs have homologous overlaps to the target on both sides of the linear donor DNA. And so if you're introducing a fluorescent protein fused to a gene of interest, you can set it up so that the donor has only 35 base pairs of homology on each side of the break, and the donor will be incorporated, bringing in that fluorescent protein gene at a fairly high frequency. This is showing the number of F1 offspring that has a GFP incorporated. And it seems to work best with fairly short overlaps. Don't want to get into a long discussion of it, but it can be used not only to add something, but to replace something. Either putting GFP, for example, in place of an entire gene, an entire coding sequence, or changing from one fluorescent protein to another using two cuts. In this method, you need to have, if you're going to do a replacement like this, you have to make two cuts in order for it to happen. With longer donors and longer homologies, you at least have the potential to do this with just a single cut. Okay, and there's a group in Japan that has done something quite similar in mammalian cells. And here one of the features that they've incorporated is that you introduce the donor on a circular plasmid, but you set it up so that the linear version of this with these short overlaps to the target is cut out at the same time that the target is cut. And you can do this by putting in, for the CRISPR version, one extra guide RNA, a guide RNA that'll cut both of those sites. At the same time, you're putting in a guide RNA that's going to cut the target. And they refer to this as microhomology mediated insertions. And you may be thinking, well, what's the difference between homology, microhomology, and no homology? Well, there's a continuum. And as we can see here, you can get not very efficient but effective insertion with quite a range of lengths of homology. Okay, so using ZFNs, TALENs, and CRISPRs, people have made all sorts of different types of genomic changes. You can, we talked about some of them. You can make translocations. Maria Jason's lab started doing this a while back by making one break on one chromosome and another break on another chromosome, and then at some frequency, a low frequency, get the translocation to occur, and they've modeled cancer translocations this way. You can make large deletions by cutting with two nucleases. This is an example where there's one break made in the CCR5 gene, and then in different experiments, additional breaks made somewhere downstream as much as 100 kb away, and you can still detect the products by PCR, even making 100 kb deletions. So you can make quite big deletions just by non-homologous end joining. Okay. So what if you're not satisfied with the way cells are processing the breaks you made? We know enough about the pathways of non-homologous end joining and homologous repair that we can start to think about manipulating them. And one of the things that people have done, including my group, is to try to inactivate one of the components of the canonical non-homologous end joining pathway called DNA ligase 4. And this is a summary of data from my lab. we found that when we just put in a donor DNA of a particular kind and look at the proportion of all new rosy mutants that come out, only about 20% of them were generated by homologous repair. But if we do this in a background where there's no DNA ligase 4, it's up closer to 2/3 of all of the mutant products. So knocking out ligase 4 is beneficial if what you want is the homologous products. And people have gone about trying to do this in other systems. I'm going to show you a couple of things involving mammalian cells. And quickly, just to say that this group in Rolf Kuhn's lab in Germany started out by using what people call a traffic light reporter. So this is a gene construct that has two inactive fluorescent protein genes. One of them can be repaired after a break here by homologous repair off a donor, leading to green fluorescence. The other is activated sometimes by end join repair that generates a frameshift mutation. So in the naive version of this, you have no green fluorescence because you have an incomplete Venus gene, and you have no red fluorescence because RFP is out of frame. So green indicates homologous repair, red indicates out of frame end join repair. And so the first thing they showed was that if you just look at end join repair, that knocking down DNA ligase 4 or another component of the end join pathway leads to a reduction in the amount of end join product. They also did this with a small molecule inhibitor of DNA ligase 4 called SCR7, which knocks down non-homologous end joining. And most effective in their hands was co-expressing a pair of adenovirus proteins that naturally inhibit DNA ligase 4 during an adenovirus infection. So I didn't know this until this paper came out, but adenoviruses, when they infect cells, they inactivate the end join pathway. Adenovirus generates linear chromosomes, and it doesn't want them concatenated because then they'd be hard to package. So it inactivates the end join pathway naturally. And from this particular adenovirus, adenovirus 4, these two proteins are the ones that are involved, and you can knock down end joining very effectively. At the same time, and I'm not going to go through this in detail, you can look at this paper. At the same time, doing these different methods of inactivating the end join pathway, the proportion of homologous repair is going up. This is SCR7, this is the adenoproteins. And SCR7 has also been used effectively in mouse embryos. And this is just an indication. You have, in this particular case, they were delivering Cas9 messenger RNA and the in vitro transcribed guide RNA mixed with SCR7 and then injected into fertilized eggs. And then they looked to see what sorts of products they were getting. And there was a template here as well. I believe this was a double-strand template. So if there was no SCR7, you got a lot of deletions and insertions and some homologous repair. But if you included SCR7, you got no non-homologous end joining and a lot of homologous repair. And this is from the Maruyama paper. There's another paper that shows this same thing. So let me just say a couple more words about SCR7. Some people have gotten it to work, and some people have not. I've heard that when you buy SCR7, that what you get is not a pure compound in the sense that a chemist would think of it, but has some, I don't know, some other maybe side products, whatever, in it. And it's not clear that the compound that's identified as SCR7 is what's active here. And the variability that people have found in trying to use SCR7 may be due to the differences in the compounds that are supplied. So that'll get sorted out eventually, I hope. Okay. Now I'm going to talk about alternatives to the Streptococcus pyogenes Cas9. There are a lot of these out there now. So if you, this is a person named Makarova and Eugene Koonen's group that's, over the years, as new data have emerged, classified the different types of CRISPR systems that have been identified in, in whole genome sequences of microbes. And we now have 5 types in 2 classes. And class 1 is the most common being types 1 and 3, involve kind of a lot of proteins, and they're much more complex than the types 2 and 5. The strep pyogenes version is a member of type 2A, and there's only a single protein that's required for DNA cleavage. In these others, it's not just one protein required for DNA cleavage. And then type 5, which we'll begin talking about shortly, also has a single protein that's required. And there are subclasses of the main classes, subtypes, and the strep pyogenes and strep thermophilus are part of the type 2A. Another member of this class, or at least of type 2, is the Cas9 from Staphylococcus aureus. This is the organism that gives you staph infections, but laboratory versions, I guess, are fairly harmless. And Feng Zhang's group, and particularly Anne Ran, looked at genome sequences to identify Cas9 proteins that were smaller than the pyogenes Cas9, which is 1,368 amino acids long. So they found a number that were a good deal shorter, and they tested them for in vitro cleavage. Now, in order to do this, they had to identify the PAMs that went with each of them, which I'm not showing you, but they did that so that they could test them in vitro. And they chose to work with the Staph aureus Cas9. They made guide RNAs using the repeat sequences from that organism and tracer sequences also from that organism. They found that guides, this is actually looking at activity in cells, that guides that were 21, 22, or 23 nucleotides long, this portion, all worked about the same, but if you made them shorter, they weren't so good. With the pyogenes Cas9, this length is typically 20, although we'll talk more about that later. So there will be a quiz on this, so take careful notes. This is a very complicated slide. Ben Kleinsteiver in Keith Jung's lab went about trying to modify the utility of the pyogenes Cas9 by insisting that it recognize alternative PAMs. And that's what this is all about. So they do a positive selection to see if the variants can cut a plasmid that contains a toxic gene. And they do a negative selection against a gene that's being selected for that carries the wild-type PAM sequence. So they make PAM variants and test to see can they cleave the new one, and do they fail to cleave the wild type. So they're trying to find variants that will recognize a new PAM. And these are just amino acid substitutions at various key positions. They didn't know when they started this work what amino acids bound the PAM, but these are right there. Okay? If you look at the wild type and you do an analysis that's based on this assay, you find that the wild type the wild-type Cas9 protein will recognize a target that has a PAM that's any nucleotide, GG, and then any of the 4 possibilities. After selection for a variant that recognizes GA instead of GG, they found this variant, the EQR, that doesn't recognize the wild-type PAM, but does a pretty good job on the NGA PAM, but it actually prefers NGAG and not so good at the other NGAs. And that's because NGAG was in the selection, okay? That was the next nucleotide over. And so the protein learned to recognize one more nucleotide as well as having a variant at this position. And there's also, they also derived one that recognizes GCG as the PAM instead of the wild type. So now we have some variants that recognize different PAMs. They also did this exercise on the Staph aureus Cas9. The wild type enzyme recognizes after 2 non-specified nucleotides G, purine, purine, T. And there's a little bit of slop in the T even. But they derived a version of the Staph aureus Cas9 that doesn't care about that G anymore. So this is just sort of building a broader library of PAM sequences so that you're not so restricted in your choice of target. An exciting development last year was the publication of a different family of nucleases related to Cas9, but in the type 5 family. This protein is called CPF1. And this paper came out of Feng Zhang's lab. Bernd Zetsche is the first author. And they looked at this protein. It was so when you're identifying CRISPR loci, you not only find these odd short sequence repeats, but you find associated proteins that always come along. And so that's how one knew that CPF1 was a CRISPR component, because its gene comes along with the repeats every time you find it in this class of organisms. And it seemed like a good candidate for Cas9-like nuclease. It's big, 1,300 amino acids, and it has a RUV-C-like collection of protein sequences. So they began working with this Cas9. It's from Francisella novicida, and they discovered a number of things that are pretty different from the standard Cas9. First of all, you only need a CRISPR RNA. You don't need a tracer RNA. So a short CRISPR RNA in conjunction with the CPF1 protein is enough to give targeted cleavage. The polarity is backwards from the Cas9 system. It's the 3' end of the CRISPR RNA, or guide RNA, that recognizes the target, and the extension that is required for binding to the protein is at the 5' end. So this little stem loop is part of what's recognized by the protein. CPF1 makes staggered cuts in the target. I didn't emphasize it, but the Pyogenes Cas9 makes mostly blunt cuts. That is, it cuts the 2 strands at the same position, although there's a little bit of slop on one of the strands. But the CPF1 makes definitive cuts 4 base pairs apart with a 5' overlap, just like a restriction enzyme would. Whether this has utility ultimately or not remains to be seen. And this Francisella CPF1 has a PAM that's TT and is on this side of the recognition sequence for the RNA. So it has a number of differences, but if you stand far enough back, it's the same type of system. It's a CRISPR type system. And this just shows cutting. And they studied in this paper a number of different features. One was, how long should the guide RNA be? Well, anything from about 18 to 22 seemed to work pretty well. After you got shorter than that, these are just in vitro assays. When you got shorter than that, then you weren't getting good cleavage. What about tolerance of mismatches? Well, mismatches make a difference, although in some positions they make more of a difference than in others. And we'll talk about specificity of CPF1 later this morning. What about activity in cells? Well, this actually shows the activity of two other CPF1s. One's called AS and the other LB, and I'm going to, in a later slide, I'm going to give you what the actual names of those organisms are. But they could see at different target sites using a T7E1 type assay that they could get quite reasonable mutagenesis. These are the indel frequencies based on analysis of that gel at quite a number of different target sequences in real cells. So the CPF1 is working. I think, frankly, the Francisella CPF1 did not work very well in cells, and so people have gone to these other relatives for tools. So if we just look at PAMs, we now have quite a range of PAM sequences that we can use, at least in planning, to target different sequences in the genome. The original Pyogenes Cas9 wanted any nucleotide and then two Gs just downstream from the RNA recognition sequence. The Jung lab developed variants of Cas9 that would recognize these. The Strep Thermophilus Cas9 naturally recognizes NN, AGAA, and with a little bit of slop in them. So that's another candidate. As I said, the Staph aureus Cas9 has this PAM that it's looking for, and a variant doesn't care about that G anymore. The AS and LB CPF1s like 3 Ts for their PAM, and the Francisella only wants 2 Ts for its PAM. So we've got quite a range, and that range can certainly be expanded by looking for Cas9 or CPF1 variants from other organisms. And just, yeah.
Just, can I just ask real quick about what's known regarding the position of the cut with respect to the PAMs in these variants? Is that
So that,
solidified?
I don't know that the Jung lab looked, if when they derived these variants, did they look to see whether the cuts were being made in the same place or not? I don't think they did that. You know, they did things that that validated the recognition of the novel PAMs, but I don't think they went and they looked precisely at how the cuts occurred. One of the things that can happen, I know with the Pyogenes Cas9, is that although, I won't remember which strand, I think the RNA-bound strand is always cut at the same place. But the displaced strand, I think, there's a little bit of slop in where that's cut. And sometimes, not always, but sometimes, and I don't know what governs this, you'll see a one nucleotide insertion as a common end-join repair product. And I think the reason for that is that when you make a staggered break that leaves a 1' 5' overhang, that can easily be filled in. You just add the one nucleotide from that recessed 3' end, and then do a blunt join to the other end. And that'll generate, if that was a one nucleotide stagger, that'll generate a one nucleotide insertion. Yeah?
Question about that. Do you have any idea, like broader statistics on the true frequency of this one base change bias? Because I think there's a little bit of a debate going on in the field between, which ranges from, oh, CRISPRs only make one nucleotide insertions and deletions to it's more broadly distributed.
It's certainly not true that with the CRISPR system you only get one nucleotide insertions. There are lots of different insertions. even though in some situations you get quite a few of these one nucleotide insertions, deletions are, as far as I'm aware, always more frequent than insertions, even when you have the one nucleotide insertion. And I've looked at kind of a lot of end join products from CRISPR cleavage, and sometimes you don't see the one nucleotide insertion at all. And I don't know why that is. I mean, how deep do you look? But, right, you know, in a sort of normal analysis. So it's variable, and I don't think we have complete control over what the variation is due to. Okay, before leaving variants on Cas9, I'm just going to talk about split Cas9. So let's say you want to use Cas9, but you don't want it to be active until you tell it to be active. So there are a variety of ways you might think about enforcing this. One of them is to break Cas9 up into two pieces and only let the two pieces come together when you're ready. And there have been several groups that have engineered this, and I'm just going to show you a couple of examples. This one came out of Feng Zhang's lab. What they did was they expressed the N-terminal portion of the pyogenase Cas9 with one protein extension and the C-terminus with another protein extension. And these two proteins will dimerize, but only in the presence of a small molecule. And when you introduce that small molecule, which is rapamycin, only then will the two halves of the protein get together. And the way they set this up had an additional clever portion to it, which is that they put NLSs on this protein, but a nuclear export signal on this protein. So this protein may be going in, but it's not doing anything in the nucleus because it's not a Cas9. But when it's synthesized in the cytoplasm, where this is hanging out and you present rapamycin, then the complex goes into the nucleus. I think it's true of all of these split Cas9s that they are not as efficient as the wild type. But if you want regulation and you're okay with adequate efficiency, not perfect efficiency, this is a reasonable way to go. And this is just an example of some target. Well, this is a target, EMX1, and then some secondary targets. And one of the things they're showing here is that these secondary sites are not cleaved and mutagenized, this is indel frequency, very effectively using the split Cas9. It's a way to regulate the activity to try to minimize some of these off-target cleavages. This is a very blurry figure, but this is another version where the halves of Cas9 are fused to portions of an intine, which is a protein that can cleave itself. It's a protein segment can cleave itself out of another protein, and when the intine segments associate, they can cleave themselves out. But the way this has been set up, it requires a guide RNA to make a conformational change in the Cas9 protein in order to get the cleavage to occur. So it's another version. And I've given you a list of a bunch of papers that were published in the last year or two. This is the rapamycin. In the Doudna lab, they made the halves a little bit differently based on structure rather than just on linear sequence. There's a photoinducible version. This is the intine-mediated one, and then a hormone-inducible one that you can look up. And my last slide for the morning is just to tell you that I try to keep track of the organisms where genome editing has been successfully performed in the real genomes of these organisms. And the list now is over 80 different organisms, and it's probably not up to date. I've added two that I know aren't published yet because I just heard about them at a meeting last month. But you can see that the phylogenetic range of organisms where these tools have been used successfully is enormous, all the way from invertebrates, including single-celled organisms. We have insects and marine organisms, vertebrates from fish to amphibians to mammals, and then lots and lots of different plants. And you're going to hear later in the week about applications to some of these different organisms. When Dan Voitas is here on Thursday, he'll talk about applications to plants. He was the first one to make use of probably each of the platforms as they came around in plants. And Scott Fahrenkrug also on Thursday will talk about applications to cows and pigs, which may be going to be even more important than applications ultimately to humans, which you'll hear about on Friday. Anyway, that's all I have for this segment. Additional questions? Yes?
Like all this RNA recognition molecules are using a ruler mechanisms where they recognize a sequence and they cut somewhere else. Does the location of PAM sequence influence as homologous versus non-homologous end-joining?
Not sure how your comment relates to the answer to the question, but so ask the question again.
So if the PAM sequence is 20 bases away versus if PAM sequence is 30 bases away from the cut, will it make a difference of choice whether it goes through homologous or whether it goes through?
I see. So, yeah, so with the Cas9s we know about, the cut is actually pretty close to the PAM. For the CPF1, it's farther away. There's sort of a broader question about whether the asymmetry of binding influences the outcome, whether it relates exactly to where the cut is or whether it relates to whether the protein might be holding on to one end at the break as opposed to the other. And people are still working on this. For the Pyogenes Cas9, if it makes a break in a test tube, it holds on to both ends really tightly. It's hard to get it off. When the Pyogenes Cas9 makes a break in cells, the cells have no trouble recognizing and processing the double-strand break. There isn't any solid evidence that it processes the two ends differently. If you look at a small collection of data, sometimes you can see, oh yeah, it looks like, downstream, or how we would look at it, upstream of the PAM, maybe the deletions are going that direction more often. But you go and you sit down with different data sets and it just doesn't, in my view, it just doesn't really hold up. So I think in cells, when the break is made, the cell detects that. How it detects it, I don't think we know precisely. But particularly in mammalian cells, there's very complex machinery that's recruited to the break. And part of the job of that machinery is to recruit the repair factors themselves. And except for minimizing the effectiveness of the end join pathway, we haven't figured out good ways to tell repair to go toward end joining or to go toward homologous repair. And people have tried overexpressing RAD51, things like that, and so far without any real success. But you may have detected that one of my key interests is how do the mechanisms that are available in cells affect what we can do? And how can we manipulate those so we can do them even better? I mean, this is what the technology is about, trying to corral the technology and make it optimal for our purposes. And it's only by understanding what's going on in the cells, it will be able to do that because the cells are determining the outcome.